cell cycle.
Yeah. So if this one gets like interrupted, or like it doesn't finish, the cell is like super. It's like it hasn't grown to like where it's ready. And then I don't know if the other thing this one, I think I wrote down the DNA, the DNA. The DNA would be like messed up because if it's like not proofreading it right.
Yeah, then like no one's carrying on.
Yeah, exactly. And then I think so.
Yeah.
All right. If you don't already have them out, pull out your unit, what is this, 8? Unit 8 notes. So we can talk for a little while about regulation of the cell cycle. As we've been talking about for the last few days, the cell cycle is kind of the life cycle of a cell where it grows and divides to make new cells. And it's really important that that cycle happens in a regulated and controlled way. Because if cells just divided whenever they felt like it, then growth would be a very weird thing, and you would have growth of tissues in places you didn't want them to be and not enough growth in other places. And so this whole process needs to be coordinated and controlled and regulated. And even tissues that have mostly finished growing, like we talked about, sometimes need to kind of start growing again if there's an injury or something like that. And so there are a lot of different things that kind of go together to tell cells when to move ahead in the cycle and when to kind of pause and stop. The first thing we're going to talk about is this idea of checkpoints. The analogy that's often used when we talk about the cell cycle, and if you read this part of your book, they use this analogy, and it's kind of a good way to get yourself to think about it, is moving through the cell cycle is kind of like the controls on a washing machine. There are places, if you've ever listened to a washing machine washing your clothes, there are places where the machine kind of pauses and stops, and you think it's done, but it's really just kind of checking, like, hey, did all the water drain out? Is it the right temperature in here? Whatever. And then it kicks itself on into the next part of the cycle. And then at the end of that part, it kind of pauses as it checks things and shifts and whatever. And just like a washing machine, the cell cycle has checkpoints like that. And it's controlled by both external and internal factors. Like I said, the washing machine itself actually has sensors that like detect the temperature and the water level and things like that inside. But it also requires you to push go on the outside to get it to start, right? So the cell cycle is kind of like that as well. There are points where the cell kind of pauses to take stock of how things are going and make sure it's ready before it goes on to the next part. And it requires certain things to be happening both inside the cell, and it responds to signals from outside the cell. So we'll kind of talk about all those aspects as we go through this today. The first is those idea of checkpoints. And you already looked at this a little, so this should hopefully be a review of that cell cycle packet. But there are 3 main checkpoints within the cell cycle where it pauses to take stock of how things are going and determine whether it's able to move on to the next phase. The first one, so a cell's life cycle kind of starts at the top at the beginning of G1 after division. You have a little tiny cell. It grows and reproduces organelles and all that kind of stuff. And then at the end of G1, there's a checkpoint that's sometimes referred to as the restriction point. This checkpoint in a lot of cells is kind of the most important one. Here, the cell is basically checking for things outside and inside the cell. It's checking to see if it's receiving signals from outside that are called growth factors. And that's something we'll talk about later. But like if you cut your skin, all of the cells around the edge of the cut will start releasing these signaling molecules called growth factors that stimulate all of the cells nearby to start dividing, to say, hey, we got some injuries here, we got to heal it up. So it'll detect whether there's growth factors. It'll detect whether it has enough nutrients in the surrounding environment and inside the cell. So these first two are kind of external things that it's checking for. This always unaligns itself for 4th hour. These are kind of external factors. It'll also check a couple of internal things. It'll check to make sure that it has grown enough to have enough cytoplasm and have enough organelles and stuff in order to divide into 2 cells. And it will also double check for damage to the DNA. If any damage to the DNA is detected at any point in the cycle, the cell will usually not go through with cell division. And sometimes it'll actually trigger that cell to go into apoptosis, which is programmed cell death. The cell will kind of self-destruct so that it doesn't pass on damaged DNA. So it checks for DNA damage. So those bottom two are kind of internal things that it's checking for. Like I said, this first checkpoint, that restriction site, is the most important of these checkpoints, because if a cell gets past that first one, it will generally move all the way through the cell cycle, which should kind of make sense, because after that, it's going to go into the S phase, where it starts replicating its DNA and actually getting ready to divide. There are a couple more checkpoints where it could abort the process if something's not going right, but most cells that pass this first point will continue through the whole cycle. Okay, so that one's probably the most important checkpoint. That's where most cells that don't divide are going to get kicked out of the cycle. And cells that aren't ready to divide, that don't meet those criteria, will often just get shunted off into that G0 phase. And they'll just kind of hang out and do their cell thing for a while until they get some signals that tell them they do need to divide. If they move past that first checkpoint, they go through the S phase and duplicate their DNA. They go through the G2 phase and kind of double check and proofread. And the next checkpoint is kind of at the end of G2 before it starts the process of mitosis. That G2M transition point checks for a couple of things. It checks again to make sure the cell is big enough to actually divide and have enough material, have enough resources, nutrients, all that kind of stuff. It again checks for DNA damage and double checks that replication is complete and as accurate as the cell can possibly do it. As we know, occasional mutations get through, but at this point, the cell is checking to make sure there aren't any significant or severe mutations or any damage to the DNA. If it meets all the criteria there, the cell's big enough and has enough resources, the DNA has all been successfully copied, and it doesn't look like there's any apparent damage, then the cell will continue into the process of mitosis. The third checkpoint happens midway through the process of mitosis. It's actually at the end of metaphase. This is the checkpoint that must be passed in order for anaphase to begin. So if you think about where the chromosomes are and what's happening at that point, during metaphase is when the chromosomes attach to the spindles at those kinetochores in the middle of the chromosomes and kind of line up in the center of the cell. And so that checkpoint won't be passed until all chromosomes are attached to the spindle on both sides. This is a way for the cell to make sure that the daughter cells, the 2 cells you end up with at the end, get the correct number of chromosomes. Because if the chromosomes weren't all attached on both sides, both sister chromatids weren't attached to the spindle before anaphase started, then 2 of those chromatids would get pulled to one side and none would go to the other, and you'd end up with 2 cells, one of which was missing a chromosome and the other one that had an extra copy. That does happen sometimes. That's what causes things like Down syndrome, and there are a few other things that's called non-disjunction, but this checkpoint is supposed to prevent that from happening. And the vast majority of the time, it stops the cell until the chromosomes are all attached. Then it allows the sister chromatids to separate and the cell to move into anaphase. Questions about those checkpoints? Okay. So we said that cells need to respond to signals in order to move through this cycle. Internal signals, external signals. Take a minute, turn and talk to somebody next to you about what we talked about years and years ago, maybe a couple months ago, about cell signaling and signal transduction pathways. What do you remember about that? What do you think of when I say those words? I know it was a while ago. I see all your eyes getting real big. Turn and talk about that for a minute and see what you can pull back out of your brain.
Is that the neuron stuff? I don't know what it's called. I don't think it's called an enzyme, but you know what I'm talking about? There was like different body systems.
Yeah.
See if this helps a little bit. Let's see. I don't know if I can draw this very well.
It like looks like an elevator. It like went on the. It would be like this kind of thing. It would come out on this side and then like this, or like something like that, right? I don't know.
Do you have a vague memory of learning about something like this a long time ago? Yes? If not, it's okay. You've got a few months to review still, right? Remember those review plans I had to make a week or two ago? This is why. So we talked a long time ago about signal transduction pathways and how cells can receive signals. So this, we call this signaling molecule a ligand. That's a little molecule that bonds to a protein receptor. The receptor could be in the cell membrane. That's how I drew it. It could be in the middle of the cell, but that's the reception of the signal. And then that, when the ligand bonds to that protein in the membrane or inside the cell, it causes that protein to change shape, which then activates another molecule, which maybe goes and phosphorylates another one, which then releases a bunch of second messenger molecules that then go out and trigger some kind of cellular response. Do you remember doing a case study called My Dog is Broken? Yeah, it was really weird and awkward because it was about erectile dysfunction in dogs. Yeah? Remember? That's partly why we do that case study, because you remember it, because it's weird and awkward. That was a case study in cell signaling, okay? And looking at how one chemical can affect these pathways and these responses. A lot of the signals that cells receive that control whether they move past checkpoints use cell signaling pathways. They're molecules that might come from, or they're signals that might come from neighboring cells, or they might be chemical signals sent out by other cells that tell a cell to start dividing. And so the response would activate some of those proteins that move it past a checkpoint in the cell cycle. Okay, so I just wanted to dig that out of your brain for a second, because this is another place where this type of signaling is really important. Okay, so there are a few different places that cells get those signals to inform them whether or not they should move through the cycle and divide. One type of signal is called density-dependent inhibition. Cells, when they reproduce, so think about something like your skin, that's a good example of this. As your skin cells reproduce, they get packed more and more closely together, right? And you're supposed to have a couple layers of skin cells, and that's it, right? So once you have enough cells to cover your entire body, those cells don't need to continue dividing immediately. So they realize that they're kind of touching other cells all around them. There are proteins on the surface of those cells and other signaling molecules that when cells physically can detect molecules of another cell membrane right up against them, that triggers one of those signal transduction pathways into the cell. And the response is, stop dividing. We have enough cells. We don't need any more right now. But if you get cut or something, all of a sudden that signal on one side of the cell is gone. And so then it activates that cell to begin dividing. Does that make sense? So that's density dependent inhibition. Cells will stop dividing when they reach a certain density. Another type of signal that we've already mentioned once or twice are growth factors. So these are chemical signals that are sent out by other cells in your body that tell cells to divide. So these can be sent out around the site of an injury or something like that. But these are chemical signals. They can also be hormone triggers that tell your body it's time to go through a growth spurt. So there are different types of chemical growth factors that tell cells to start dividing. Most tissue types in your body also exhibit something called anchorage dependence. Think of being anchored as being like tied to the ground, right? Most cells have to be attached to something, to a substrate, which just means a surface below them, or the extracellular matrix, that's ECM. If you remember when we learned about cells, there is this like matrix of fibers between all of the cells in your body. We don't have cell walls, but there is still this network of filaments outside of your cells. If cells aren't attached to something, most cells will not divide. Yep. So all of these are examples of external signals that can trigger those signal transduction pathways. And the response at the end of those pathways usually is to activate these molecules called cyclins. You looked at some of these in that packet as well. The main groups of molecules that tell cells to move past checkpoints are cyclins and kinases, which are two different groups of protein molecules in your cells. You may recognize kinases. We've talked about those a few times. And when we talked about signal transduction, kinases are the molecules that will phosphorylate other molecules. And when you phosphorylate something, that means add phosphate to it. That's a way to activate a molecule because a phosphate group changes the shape, the conformation of a protein, and usually works to activate it. So as part of those signal transduction cascades, these kinases phosphorylate and activate proteins called cyclins, whose main function is to regulate the cell cycle. Hence the name cyclin. And so it's a combination of all those signals coming from outside the cell and checking on the DNA and those chemical signals coming from inside the cell that determine whether those cyclins become activated to tell the cell to move past the checkpoint and continue with the cell cycle. Questions at this point? Okay. Sometimes all of these very complicated chemical controls don't work. This is something we're just going to touch on briefly today. If we have time towards the end of this unit, we'll probably go a little more in depth into this because it kind of comes back later in the unit as well. But if these controls that are supposed to regulate when the cell moves forward and when it stops moving through the cell cycle, if these don't work, then those cells can become cancerous because they begin dividing and dividing uncontrollably. They're not responding to those stop signals that tell them when to stop dividing, and you end up with this pile up of cells in a place it's not supposed to be, and that's what a tumor is. And depending on the type of tumor, if it breaks off and moves somewhere else, that's a malignant tumor that's starting to travel around your body and get really dangerous. So we're going to actually watch a video. It's about 12 minutes that will talk a little bit about this. And as you watch this video, I want you to take some notes on the terms listed on this slide. So you have a basic understanding of what they are and their role in controlling the cell cycle when it's healthy or causing cancer when it's not. Okay? Before I put this video up, I just want to talk about one next thing, because then I'm going to let the people at home go watch this video on their own if they'd prefer. After the video, I was going to wait until tomorrow to post this, but I realized we might have some extra time today, which we do. So tomorrow we're going to be doing a mitosis lab, which will mainly involve looking at slides of cells going through mitosis and practicing identifying phases like you probably did in 10th grade, with some statistical analysis in there as well. This is probably the biggest lab we have left this trimester. It's not super intensive with what we're doing in class, but there is a lot of statistics. Part of the point of this lab is to review things like chi-square and standard error of the mean. Remember those error bars that we drew on graphs a long time ago? And remember some of that stuff that you learned way back in first trimester? So I will post that as soon as we're done watching this video here. If you want to start on it now, there's actually quite a bit that you could do before we actually collect data in class. If you really, really don't remember statistics, the post for this lab, I actually attached a couple videos to review those statistical tests. So if tonight you just want to watch those videos so that you remember what that's all about, you can. And you can also start on part of the lab that does not require the in-class data. Okay, so I'm going to post that today if you want to get a little bit of a jump start on it. I'm going to give you like a week to do this lab, so if you have questions, you can ask. But for those of you that don't like wasting time, we're going to have some time left today. So I'll put that up so you can start working on it. Okay? Okay. People at home, I will put the link to this video in the chat here in just a minute. And then if you would prefer to just go watch this video on your own, you can click on that link and then you are welcome to log out of the meet. Just please make sure you take some notes on those terms on the previous slide as you watch the video. And then, like I said, I'll post that mitosis lab. If you want to start looking at it today, we will talk about that a bunch more tomorrow. Okay? Okay, give me 1 second.